Without further ado, let me introduce you to Dr. Bruce Korf, who is the Chief Genomics Officer for UAB Medicine and the Director of the Heersink School of Medicine Neurofibromatosis Program and Clinic. Dr. Korf is the Chair of Medical Advisory Board. He is of the Children's Tumor Foundation and the Principal Investigator for the Department of Defense funded Neurofibromatosis Clinical Trials Consortium. So Dr. Korf, take it away.
All right, thank you. Actually, I've retired as chair of the Medical Advisory Board of CTF after 33 years in that role, but I'm still very much involved. So first of all, thank you, Rini, for organizing this and all the hard work that I know goes into that. And thanks to everybody who's here for attending. As always, we, on the one hand, wish that we could be in person. That's how this symposium always had occurred over the years until, of course, the last few. But the good side of it being virtual is that, as you heard, we have people from all over the world able to attend. And so, you know, that's something which we'll have to figure out a way of accommodating that even in future years when we do hope that we'll be able to get together in person again. Well, so you're going to be hearing from many people in the program, as you've just heard in the agenda. What I'm going to try to do is summarize a few things that are new in the field of NF. I want to make a few comments about the NF Clinical Trials Consortium, introduce you to the sort of breadth of the UAB NF program, both clinical and research activities, say a few words about the work that we're doing at UAB on development of novel therapeutics, and then end with some kind of thoughts about where things may be going in the coming years. Well, as I think is known to everybody on the symposium at this point, there are many different forms of neurofibromatosis. The nomenclature that we've been using for a long time is NF1, NF2, and then schwannomatosis. NF1 is the most common of the three. It's about 1 in 3,000 in the population. And this is true anywhere in the world. And is characterized by the cafe au lait spots and the skin neurofibromas, as well as other things you'll be hearing about. The gene has been known for 32 years now and is a gene that encodes a protein called neurofibromin. The gene is located on chromosome 17. NF2 is characterized chiefly by tumors of the hearing nerves. This is the MRI scan, and here are the hearing nerves on each side, and this is pretty obvious tumor on this side, and other tumors that occur in various parts of the nervous system. The tumors of NF2 principally are called schwannomas, and there are other tumors, for example, meningiomas, which involve the covering of the brain and spinal cord, and ependymomas, which occur usually within the spinal cord, would be the most common place in NF2. And then the third condition is schwannomatosis, where you get tumors that are schwannomas, somewhat similar to the schwannomas you see in NF2, that can occur in various places and usually cause pain. And the genes involved are quite different. The gene for NF2, well, it's called NF2 and encodes a protein that has come to be called Merlin. It's located on chromosome 22. And so far we know of two genes associated with schwannomatosis. They're both also on chromosome 22, all clustered together, actually. One's called LZTR1, the other is called SMART-B1. So you might wonder why all of these are called neurofibromatoses, even though actually only NF1 produces neurofibromas, and NF2 and schwannomatosis actually are associated with schwannomas, not neurofibromas, and you may wonder what's the difference between these. So here's a admittedly crudely drawn diagram that may give you a window on what the difference is. So what you're looking at is a nerve, and the little fibers in the nerve are individual nerve fibers. You can think of a nerve as like a bundle of wires. And in a neurofibroma, there's a diffuse growth where the nerve fibers themselves are enlarged and fill the space that comprises the tumor. Whereas in a schwannoma, there's actually the nerve fibers are running along, and there's a proliferation of Schwann cells that push on the nerve fiber. So the nerve fiber itself runs all through it, but then there's this associated tumor. Now, they both derive from Schwann cells, and Schwann cells are the insulating cells around the nerve. Think of a nerve similar to a wire with an insulation surrounding it, and that insulation is principally made up of Schwann cells. But neurofibromas and schwannomas are actually quite different, and are tumors that often cause different symptoms even. Neurofibromas usually don't substantially impair the function of the nerve unless they get big enough that they press on a surrounding structure and compress the nerve. Schwannomas can compress the nerve and are actually more likely to cause pain. So why is NF2 and schwannomatosis considered a neurofibromatosis, even though they don't produce neurofibromas? Honestly, the reason is that when these conditions were first studied, which now dates back more than 100 years, all people really could tell was that these were conditions that involved tumors of the nerve sheath, and wasn't easy to distinguish the details. And it wasn't really until about, I'd say, the 1960s that the difference between NF1 and what then came to be called NF2 was discerned. And then it wasn't until the 1980s and into 1990 that the genetics were discovered to be different. So there has been an international effort to address this, because you could argue it doesn't make a whole lot of sense to name a condition neurofibromatosis type 2 and they don't get neurofibromas. And there is an ongoing effort to kind of describe NF2 and schwannomatosis as what are called schwannoma predisposition syndromes, or schwannoma predisposition syndromes, or SPS, and then distinguish them as SPS NF2, and it's what we now call NF2, and SPS LZTR1 or SPS SMARCB1. And then there are some where you can't find the gene, and those would be kind of not otherwise specified. So there's a lot of work going on in the background, and there'll be more probably to say about this, and updated diagnostic criteria that I expect are going to be forthcoming very soon. So a bit premature to talk more about those right now, but there is quite a bit more to come, I think, very likely in just the next few months. The manifestations of NF1 do tend to evolve with time, and although nerve sheath tumors are the hallmark, as you can see, lots of other things happen. Usually very early in life, head size will be larger than average, although this is just due to a larger brain size itself. It doesn't seem to cause any problems. We'll tell you a little bit later about plexiform neurofibromas, but we think those are present from early in life, maybe even from birth. Cafe au lait spots, the brown spots that are characteristic for NF1, usually are visible by the first 2 years of life. If there's going to be a problem in bone growth, it usually happens, I would say, even by the time a child is able to bear weight, and usually within the first year. During childhood years, curvature of the spine or scoliosis. We're vigilant for optic pathway tumors in children, and usually if those are going to happen, they will happen by about age 6 or so. Skin fold freckling under the arms and in the groin region by about 3 to 5 years of age. A small proportion develop a kind of narrowing of the artery to the kidney, which can cause high blood pressure. So we keep an eye out for that. Narrowing of arteries to the brain, which produces a condition called moya moya. Not very common, but when it happens, it can predispose to strokes, and that's another thing to be vigilant for. And very commonly, you'll hear more about this later, learning disabilities, attention deficit disorder, and the like. And then in adults, a few things that might not be widely recognized. Glomus tumors are tumors in the nail beds, finger and toenails, and they can cause really severe pain when either there's pressure on the nail or if they're exposed to cold temperatures. And a lot of people don't realize these are part of NF, and they're actually very treatable surgically. Malignancy is, of course, always a concern, and there's about a 10% lifetime risk of a malignant peripheral nerve sheath tumor. I'll say more about that in a bit. People with NF1 often have osteoporosis, bones that are undermineralized, and we are now routinely checking vitamin D levels, although I can't tell you that treating the vitamin D entirely prevents this, but it is an important thing to watch for. Pheochromocytoma is a tumor of the adrenal gland and causes episodes of high blood pressure and heart racing and sweating. Neurofibromas usually begin on the skin somewhere in the adolescent years, although you can see them earlier and never can predict how many there are going to be. And many women notice they get more when they're pregnant. We now know that women with NF1 are at increased risk of breast cancer and recommend mammography screening starting at age 30. And although you don't hear much about this, lung cysts and also pulmonary hypertension, problems with blood flow in the lungs, are known to occur in some people with NF1 in adulthood. NF2, mostly we're dealing with tumors of the meninges called meningiomas, which can, though benign, can cause pressure on the brain or spine. Vestibular schwannomas are the schwannomas that occur on the hearing nerve and very often cause loss of hearing. It can be schwannomas of other cranial nerves. Ependymoma, a tumor of the spinal cord most of the time, very often pretty benign and not always requiring treatment. There can be schwannomas of the nerve roots coming off the spine. There actually can be schwannomas in the skin, usually benign and, well, actually always benign, but usually not causing major problems. And something you don't hear much about, but many people with NF2 develop a peripheral neuropathy, will develop weakness and numbness and tingling. And then schwannomatosis, so far as we know, mainly causes schwannomas. Some forms can cause meningiomas, but for the most part, schwannomas and pain. And the pain is often out of proportion with the size of the tumor and tends to be relieved by removing the tumor, although that's not always so feasible to do. These are all transmitted as dominant traits. So an affected individual is filled in symbol, females are circles, males are squares. And these all go from generation to generation. There's no difference in transmission, whether it's a female or male transmitting or a female or male child. And risks to anybody who has a genetic mutation for any of these genes is 50-50 of passing it on to any of their children. And NF1 and NF2 do not skip generations. So if a person carries the gene, you can pretty well count on the fact that they're going to show some signs. I'll give you one caveat to that in a minute. Half the time, though, when we see a person with any of the forms of neurofibromatosis, they're the first affected in the family due to a new genetic mutation that occurred in the sperm or egg that formed them for no reason that we know of. These are random events. They're not due to exposure to chemicals or radiation and really are unpredictable. Now, schwannomatosis is perhaps the one exception because there are people who seem to carry the gene and show fewer no signs. And you'll see why I think that is happening in just a little while. So what's new? There are newly revised diagnostic criteria for NF1. And actually, there are newly revised diagnostic criteria for NF2 and schwannomatosis, but they aren't yet released. And I think that, as I mentioned, will happen very shortly. So I won't talk about those yet today. They're relatively small but important changes that should make diagnosis easier. But the NF1 diagnostic criteria were actually released now something about a year ago, actually, as you can see from the paper that was published. And they're still, and I think Dr. Neighbors will talk some about this too, mostly the same: cafe au lait macules, skin fold freckling, neurofibromas, optic glioma. I put in red the two changes. One is that if a person has either Lisch nodules, and I'll show you what those are in a minute, or choroidal abnormalities, you don't have to have both, but one or the other would constitute a diagnostic criterion. And now they've added the possibility of a what's called non-mosaic, and you'll see what that means in a minute, pathogenic NF1 variant. And this would all be for a person without a family history, and any two criteria fulfilled makes a diagnosis of NF1, with the exception that cafe au lait spots and skin fold freckling, if that's all that's present, can also be a sign of a related condition called Legius syndrome, and that can only be distinguished from NF1 if that's the only sign present by genetic testing. Now, if a child has a parent who meets the criteria, then the child only needs to fulfill one criterion. Since I mentioned the eye changes, this is an example of Lisch nodules. You're looking at a close-up of the iris of the eye, and you can see these are kind of tan bumps on the iris. They actually are accretions of pigment cells that grow. They're harmless. They don't seem to affect vision, and they're pretty reliably present in people with NF1 after about age 6 or 7. You can't really see them well with the unaided eye, and even an ophthalmoscope, which is what typically we use in an office to visualize the eye, doesn't distinguish these reliably from other kinds of pigmentary changes in the iris. An ophthalmologist uses a special microscope called the slit lamp, and that shows these to be, as you can see here, 3-dimensional bumps on the iris. Okay, then optic glioma, this is the tumor of the optic pathway. This is an MRI scan of the brain. You're looking in kind of cross-section, and you can see the enlargement of both optic nerves. And when you're looking in a side view, this is showing an enlargement of the optic chiasm where the 2 optic nerves meet just in front of the brain. And this is not rare in NF1. About 15% of children will have it. Only the majority do not have symptoms and don't need to be treated, although it can cause loss of vision and problems with hormone secretion, usually causing precocious puberty. Those are not common, but definitely important things to watch for. And when that happens, then various treatments are needed. Here's a picture I pulled from a paper that's referenced down here of the choroidal abnormalities, these little white patches that you can see in the retina that an ophthalmologist will see and may need to use special tools, which are pretty routine in ophthalmology care now, if the ophthalmologist knows that they're looking for this. And these are also generally harmless, but they have been accepted now as a diagnostic criterion for NF1. And it can be helpful to look for these if there's some doubt about the diagnosis, and can serve as an additional clue that a person is affected. Well, as I mentioned, genetic testing was added to the diagnostic criteria. And now genetic testing for NF1 has been available since, well, actually in a way since the gene was discovered in 1990, but I think it became fairly routine to do in about 2003. And actually that was when Ludwig Messiaen joined the faculty at UAB, and she had really developed the first practical clinical NF1 test. And this is a slide which you can see is only a few years after she started doing the testing. And all the little asterisks below a diagram of the gene show where different mutations have been found in different people with NF1. You can see they're scattered all over the gene. And it's a pretty big gene. And then these lines are deletions of chunks of the gene, and the vertical lettering are mutations that affect the way the gene is processed in the cell. So the mutations are all over the place. And it's one of the things that probably explains why mutations occur pretty commonly, because the gene is a pretty big target. And most of the mutations impair function of the gene, and doesn't really matter where they are. If they turn the gene off, the outcome is basically going to be the same. Just to remind you of what a gene actually is, you're looking at a diagram of the double helix of DNA. The genetic code are these 4 letters, G, C, A, and T, and they spell out the recipe to make proteins. And the words of the genetic code are 3 letters, so G, C, A here tells the cell to put an amino acid, which is a building block of a protein, called alanine. And then the next one here is AGA, and that spells arginine and tells the cell to put an arginine next to the alanine. And each protein has a unique sequence of amino acids determined by the sequence of letters of the genetic code. And a change of even a single letter can be very significant to the cell, and can cause either the wrong amino acid to be put in the protein, or sometimes to cause the cell to stop making the protein entirely. And when this happens, it can cause significant problems. I illustrate this with this kind of simple example of a sentence of 3-letter words, and you can see how a single change, like for example, changing the D to an H, says the boy pet his hog, which is obviously not what happened. And so a single letter change can make a big difference in the meaning of the sentence. Or you can lose a word. Here, the 3 letters were lost, and you have the boy, his dog, also makes no sense. Or you can insert something, which likewise makes no sense. And then there's something called a frame shift. And in this case, you add something here that doesn't belong here. And, or I'm sorry, in this case, we deleted something. So we took out the Y, and you resort the 3 letters, and you end up with a sentence that makes no sense at all. And the same would occur if you add something that doesn't belong. You kind of assort these 3 letters, and it produces complete nonsense. So a single change can have devastating effect, even though it's only 1 letter out of over 300,000 that comprise the NF gene. People often ask, if you do genetic testing, can you predict how severe NF will be or what particular manifestations? The answer is, for the most part, no, but there are a few exceptions. People who are missing their entire gene, we call that a gene deletion, usually have a pretty severe set of manifestations. They get their neurofibromas at a young age, and lots of them. They seem to be at about twice the risk of malignant peripheral nerve sheath tumor as those with other mutations. They tend to be tall, where people with NF1 are usually short. And they usually have more significant intellectual disability than you otherwise see. And they frankly just look physically a little different. Most of the time when somebody comes into clinic with a large deletion, it's pretty obvious if you've seen a lot of people with NF. There's a few others. I won't go through the details here. There's a specific mutation that produces some unusual facial features and cardiac malformation called pulmonic stenosis. There's a cluster of mutations that produce pretty severe manifestations. On the other hand, there's a particular mutation where there's a deletion of 3 bases, which deletes 1 amino acid. And in that case, you don't get neurofibromas or tumors, although you do get learning disabilities. And then another cluster of mutations that produce cardiovascular problems, as well as spinal neurofibromas. And there's some evidence that when the mutations cluster near one end of the gene, there may be an increased risk of autism spectrum disorder and brain tumors. One of the things that we see not so rarely is a phenomenon of mosaicism. And what that means is that in general, a person who has NF1, NF2, or schwannomatosis is born with every cell in the body carrying the mutation, because that mutation was there in the sperm or egg that formed them. That could either be because it was passed on from a parent, or the mutation occurred when sperm or egg cells were made. Either way, from the moment of conception, the mutation is there. However, there are some instances where the early embryo, which you can conceptualize as a cluster of cells, one cell undergoes mutation, which is depicted here. And then what you end up with is a mixture of cells, most without the mutation, but some with it. And the outcome later on is the person might have patches of their body with various of the features of NF. And if it's NF1, we tend to call that segmental NF, where we see maybe a cluster of cafe au lait spots and freckles just in one region and not in others. It can really vary in terms of where it is. Sometimes it can cause just one isolated feature of NF, like a plexiform neurofibroma, or a vestibular schwannoma, or even a meningioma. There can be a phenomenon of gonadal mosaicism, where the only cells involved are in the germline, and the parent is free of signs, but has more than one sperm or egg cell with a mutation, which means they can pass it on to more than one child. That's pretty rare, but it's been known to happen. And we've learned that many people with NF2 or schwannomatosis are mosaic. So we find that they have 1 or 2 vestibular schwannomas, they have maybe meningiomas, or they just might have a few schwannomas. And we test their blood and we can't find anything. And the only way you can determine the presence of a mutation is by testing tumor tissue. And the lab at UAB is set up to do that right now on NF2 and schwannomatosis. And actually, pre-COVID, we were able to culture cells from neurofibromas and even from cafe au lait spots in people with mosaicism to find the mutation. That's a pretty challenging and labor-intensive approach. And in the COVID era, because of staffing issues that had to be temporarily curtailed, I know the lab is working to reinstate that testing, and I hope that will be back online before too long. All right, let me switch now and talk for a minute about the NF Clinical Trials Consortium, and then we'll talk a little bit about treatment. Actually, dating back to about 2008, the Department of Defense, through the Congressionally Directed Medical Research Program, has funded a clinical trials consortium that is charged to do clinical trials for all forms of NF, NF1, NF2, and schwannomatosis. In its first iteration, there were 9 sites that are shown in red here. And then in 2011, we added 4 new sites, and in 2017, 2 sites in addition. Plus, many of the sites have affiliate sites. For example, the UAB site has an affiliate site in Atlanta at Emory. And actually, the consortium was up for competitive renewal just in the last few months, and we submitted our proposal at the end, or in the fall, I guess, of last year, and it has been selected for funding. We're just in the process now of working with the DoD to organize the contracts for the consortium, and the new funding mechanism should begin in August of this year. The previous 3 iterations were 5-year funding for the consortium, and this one is actually 10 years of funding, with the DoD now playing a kind of critical role in working with us. UAB, as I'll say again in a few minutes, is the hub of the consortium. We're the coordinating center, and all of these sites are involved in enrollment of individuals for clinical trials, as well as generation of protocols for clinical trials. And this is a diagram showing you the clinical trials the consortium has done since its inception. Most have been NF1, but a few have been NF2 focused. We have not yet launched a schwannomatosis trial. We actually worked on one for quite some while. It proved to be just for logistical reasons difficult to implement. But here are the 3 funding cycles so far. And these are the trials that the consortium has done on its own, which you can see there's quite a number. And then there's a number of collaborations that we established, especially with a sarcoma alliance for our malignant peripheral nerve sheath tumor trials. And there are several that are shown up here, which are actually either soon to be launched or recently launched. NF111, which is a low-grade glioma trial, is now active, and these other trials are in process. They haven't yet been launched, but should be within the next year or so. And you can go to clinicaltrials.gov, which is a website that is a kind of index of all clinical trials registered with the government, and find out the details of any of these clinical trials and what enrollment sites are involved. Well, let me now say something about the UAB NF program, and you'll be hearing from many of the colleagues involved in the program during the course of today, but I thought I would give you an overview of how things are organized here. So we include here NF clinics, and I'll tell you in a second which these are: genetic testing, clinical trials, and laboratory research. Now, the clinics include, first of all, a genetics clinic, which I run together with our nurse practitioner, Tammy Skelton, and we also include genetic counseling within the program. And Bryce Brown will be joining us in the clinic, I think, over the course of this next couple of months. We also have genetic counseling assistants that work with us in the clinic. Our genetics clinics for NF take place on Thursdays at the Whitaker Clinic at UAB, and every other week at Children's of Alabama on Tuesday afternoons. We see children and adults with all forms of NF. We only see children at Children's of Alabama, but we can see children and adults at the Whitaker Clinic at UAB. So the appointments are made through the Department of Genetics. Now, we work very closely with both adult and pediatric neuro-oncology, and you'll be hearing from Dr. Bert Neighbors, who's the head of adult neuro-oncology, and from Dr. Katie Metrock, who's principal person seeing NF patients in pediatric neuro-oncology at Children's of Alabama. And they each have multiple staff members working with them, and they can, I think, introduce you to the team that they work with when they speak a little bit later on. So we tend to refer patients, either children or adults, either who have optic gliomas, that's usually children, to be referred to the pediatric neuro-oncology team, or any kind of glioma, which is a tumor in the brain, either to the adult or pediatric neuro-oncology team. And now that we have the ability to institute therapy, we've been referring mostly children with plexiform neurofibromas to Dr. Metrock for treatment with MEK inhibitors, such as selumetinib, for example. I mentioned the genetic testing lab was established in about 2003. And when it was established, it was the only lab in the world that was doing comprehensive NF1 testing. And then it picked up NF2 testing. And in fact, it discovered the LZTR1 gene is associated with schwannomatosis. So it's got a long and I think really renowned history. Dr. Messiaen, who established the lab, retired last year, and Dr. Yulong Fu now is the director of the UAB Medical Genomics Lab, continuing the full range of testing for NF1, NF2, and schwannomatosis. We have active clinical trials. I just told you about the NF Clinical Trials Consortium, which, as I mentioned, we are the coordinating center for, as well as a recruitment site for children and adults with various of the trials. And both the adult and pediatric neuro-oncology groups are very much involved in those trials. We have had some clinical trials that we do just individually at UAB. In addition, some of these are not consortium trials. And then we have an active laboratory research program, and I'll tell you more about that in just a moment. The principal investigators are Dr. Robert Kesterson, who's an expert in making animal models, and particularly mouse and rat models. And you'll see in a few minutes some of the work that he and his group have been doing. Dr. Deanne Wallace, who's an associate professor in the Department of Genetics, has been creating model systems, cell culture models, and doing drug discovery. And I'll explain a little bit later what in particular she's working on. Dr. Andre Lyer is focusing on a particular approach to treatment based on RNA editing, and we'll mention that very shortly. And Dr. David Bedwell is an investigator at UAB who had really discovered an approach to treatment of specific kinds of mutations. And I'll say more about that in a second. Key in terms of NF1 treatment up to this point in time, I see somebody's raised their hand. I'm going to probably, if it's okay, take questions right at the end. Is NF1 is a regulator of a protein called RAS. RAS is a cell membrane associated protein. And when a message is received at the cell surface to tell a cell to grow, it activates RAS. And when that happens, RAS is bound to a molecule called GTP, and it signals through many different pathways to promote cell growth or cell survival. Neurofibromin, the product of the NF1 gene, is involved in turning this off by stimulating the conversion of GTP to GDP, and that terminates the signal. So you can imagine when you're missing this, RAS gets stuck in this activated state and it causes uncontrolled cell growth. We've learned that NF1 is a tumor suppressor gene. What that means is that a person with NF1 is born with one mutated copy, which is indicated by the minus, but one non-mutated copy indicated by the plus. But it appears that there's just random loss of the other non-mutated copy in some cells just naturally as cells grow. And ordinarily, you wouldn't really know this because you've got a backup copy, you might say. But if you're missing that backup copy to begin with because of being born with a mutation, then the cell has no copy, and that leads to the production of a neurofibroma and the growth of cells that surround it due to signaling. And we've learned that malignancy seems to be the consequence of accumulation of other genetic changes. And this same mechanism actually applies to NF2, just change the name of the gene. And so in order to get a tumor, you really have to lose both copies of the gene. And that's a natural phenomenon, but becomes problematic when you're born with one copy not present. A lot's been learned about the way neurofibromas form. If you look at skin neurofibromas at the genetic level, all you see is they're missing both copies of NF1. That seems to be enough to cause a skin neurofibroma. Plexiform neurofibromas, same thing. And we think the difference between these is that the loss of the second copy of the NF1 gene occurs during early development, and so it affects a much larger region of a nerve, whereas these must be occurring later on. Then there are neurofibromas called atypical neurofibromas. They show up as these very, almost spherical kind of masses, and they can often be painful. And we think they are kind of precursor to possible malignant change. And if you look genetically, they're missing NF1, but they also have mutations of a gene called CDKN2A. And then if you look at malignant peripheral nerve sheath tumors, MRI-wise, they don't look that different from atypical neurofibromas, but they have much more active cell growth, and they tend to accumulate many other genetic changes on top of the loss of NF1 and CDKN2A. I'll say, by the way, that malignant change seems to be unique to NF1. It does not occur to a significant extent either in NF2 or in schwannomatosis. And in fact, schwannomatosis has a more complicated genetic mechanism. People who have a germline mutation, for example, of SMARCB1, remember that SMARCB1, and for that matter, LZTR1, are clustered very near NF2. So this is the mutation a person with schwannomatosis might be born with. If you look at the tumors, though, you find, well, they do have a SMARCB1 mutation, the one they were born with, but they've also acquired an NF2 mutation, and usually the other copy of the gene is deleted, taking out both SMARCB1 and NF2. So it's really a multiple step process. You have to lose NF2 and delete the other copy. And probably that's why the schwannomas are less frequent in schwannomatosis than they are in NF2, because it's more complicated to get a tumor. And it's also probably why some people with schwannomatosis may not get any schwannomas, or maybe just one or two, and might not ever get diagnosed. So it could seem to skip a generation, where that just never happens in NF1 or NF2. And if you have the original mutation in LZTR1, to substitute that for SMARCB1, you get the same mechanism. Well, the big news, which is not really news even anymore, and I think I spoke about it last year, in treatment for NF1 is the advent of the MEK inhibitors. And this is another cartoon showing the mechanism of NF1, which I talked about a little bit ago. And it's a bit simplified. It shows just one branch of the pathway, but one of the way stations from RAS to the nucleus, sort of like a domino effect, is called MEK. And a variety of drugs called MEK inhibitors are found to inhibit this. And first with mouse models, but then in clinical trials, it was demonstrated that these MEK inhibitors have activity to inhibit the growth of plexiform neurofibromas. I know Dr. Metrock will talk more about this, but it resulted now about 2 years ago in FDA approval of for a drug called selumetinib, goes by the brand name Koselugo, as a treatment for plexiform neurofibromas in children. And I'll tell you, we're using that now very frequently, although it's a complicated thought process to decide when and whether to treat based on trying to balance the potential for controlling growth of a plexiform neurofibroma with potential side effects. I think Dr. Metrock may talk more about that. Now, there are lots of other theoretical ways you could treat NF. So I mentioned that signaling between the Schwann cells and neighboring cells occur, and there are drugs now that can inhibit that signaling. Inhibiting RAS, well, that's what MEK inhibitors do. We've been interested here in whether we can turn the mutated gene or gene product back on. Be great if you could prevent those second hit mutations, although I don't think we've come up with a good idea of how to do it. If you look at the different kinds of mutations that occur in the NF1 gene, remember I showed you that diagram with, it's actually thousands of different mutations, but they can be classified. Some of them cause the wrong amino acid. We call that a missense. Some cause the protein to stop in its tracks in terms of being produced. We call that a nonsense. Remember the frame shift where you insert one or take away one base from the genetic code. There can be what are called copy number changes. Splicing mutations I didn't talk about, but they involve processing of the gene, or the whole gene deletions, where I mentioned you get a very severe form of NF. Well, there's different kinds of treatments that we're experimenting with. For missense mutations, what are called correctors or potentiators, drugs that would prevent the abnormal protein from being degraded in the cell, as it tends to be, or to being just non-functional. Read-through treatments, I'll say more about in a second. Something called exon skipping is being looked at. Things that can modulate and correct defects in splicing. The only way to fix a whole gene deletion is to replace the gene, and we're working on that too, either with gene editing or gene replacement. I won't go into details here, but Dr. Kesterson's lab, this is a diagram of the gene, has been creating mouse models with human mutations, many of which are shown down here, so that we can test these different kinds of approaches to therapeutics. Dr. Wallace's lab has been generating induced pluripotent stem cell cultures, where you can take cells, including either embryonic cells, which we're not working on, or fibroblast cells or blood cells, which we are working on, where you actually take these and using a variety of molecular techniques, you can convert them back into stem cells and then generate cultures. And we've done that where we can generate Schwann cells, or we also have experimented with fibroblasts. And this allows us to do drug testing on tissue that is actually not necessarily even obtained from NF patients, but we can edit the gene using the CRISPR technology and create any mutation we want. The nonsense suppression, well, a nonsense mutation causes like a stop sign to be put in the middle of the gene, and protein never gets made all the way. And there are drugs, including what are called aminoglycoside antibiotics, that trick the cell into running the stop sign and producing a full-length protein. And this is something Dr. Bedwell actually originated and is working on to find drugs that specifically accomplish this for the NF1 gene. We're doing a lot of work with missense mutations. I won't show you the full details. It gets pretty technical, but to try to find drugs that will restore function in the presence of a missense mutation. And then sometimes you can take a segment of the gene, because genes are encoded in segments, and actually the intervening sequences are spliced out, and then the coding sequence is knitted together normally. And if there's a mutation, some of the time, the exon, which is what this coding sequence is called, can be, the protein can be functional even without that segment. And there are ways of tricking the cell into skipping that exon and producing a full-length protein. It doesn't turn out that you can do that with very many exons in the NF1 gene, but we have found a few where you can, and we're experimenting with that. Dr. Kesterson's group is collaborating with a group at Yale, creating nanoparticles. Those are little particles that can incorporate a piece of DNA or RNA or other molecules, get into the cell, and they're packaging either a kind of a small version of the NF1 gene called the cDNA to replace that gene in cells, or the CRISPR technology to edit the gene, which is a pretty challenging thing. But they've got now some mouse and rat models where they're working on it. By the way, nanoparticles might sound exotic, but that's actually basically what is used in many of the COVID vaccines now. So this is an established technology which Dr. Kesterson's group is using to see if they can introduce a replacement for the NF1 gene in cells. Ribozyme's a little bit complicated, but just to give you a kind of simple version, in order to get from DNA to a protein, the gene is copied into something called messenger RNA, and that's actually what carries the genetic code into the cell so it can make a protein. And there are ways, I won't go into any of the technical details, to edit a messenger RNA and replace a mutated version with a non-mutated version. And Dr. Lyer is working on technology to try to do that to correct NF mutations. I'll just mention, I've shown you a few of the people who are involved, but it's a pretty large group, and not all of them even are pictured here, although many people are named. So the group in the NF research program at UAB has grown quite a bit, and funding from a variety of different sources that are shown below. Very close to the end of my time, just want to remind you that, it turns out, I just looked it up, started in 2014. So for 8 years now, we've been doing a blog. I won't say exactly monthly, comes close to every month. That's been the goal, where I meet with a writer who interviews me and translates that into something which gets posted on the NF program website. I try to highlight either things going on in the UAB NF program or address issues in NF that I thought would be of interest. And actually, one thing to look forward to in the coming months is the writer is going to be interviewing some of the other people involved in the program who I've just mentioned. Well, where are we going in the future? A lot of the work I've described are aimed at either smart surveillance or targeted treatment. Can we predict who's going to get what complication, either by their NF mutation or other kinds of genetic studies that are now possible, at least, hoping to find why NF differs so much from one person to the next? And can we individualize the surveillance so that it's targeted at a particular risk profile for that individual? We're not there yet, but that's, I think, where things will head in the next few years. And to what extent can we personalize treatment targeting the particular mutation a given person has? And I would emphasize the importance of community building. You heard about the Children's Tumor Foundation earlier. They have a NF registry that I would encourage you to look into, and a lot of activity for patient support, trying to collect data across multiple communities, and increasingly access, which especially these days, as we've learned how to use telemedicine, I hope will continue to increase. So with that, I am going to end, and I realize I'm right at the end of my time too. I'll stop sharing my screen.